If you want to have a full understanding of today's topic, watch our previous embryology videos. So we continue our meiosis lectures. Today we'll do the meiosis 1. Meiosis 1. And this meiosis 1, just like the mitosis, it has the 4 phases. We have the prophase. We have the metaphase, we have the anaphase, and we have the telophase. So in this prophase, listen attentively because you can easily get confused here. So assuming this is a cell, it has a nucleus, and inside this nucleus we have different set of chromosomes. So this is how the chromosomes look like. So at the beginning of this prophase, this chromosome will now coil and expand and become duplicate. The DNA will become duplicate. You see here they are just one. In this place, they are just like this. Like this. So when the prophase started, they will become like this. So do you get me up to this point? Okay. So then during this prophase, during this prophase, they become duplicate, and then they have now become double chromatids. Since if they become two, here the center is what we call the centromere, and here one of it is chromatid and the other one is chromatid. The whole thing is called the chromosome. So in writing genotypes or anything, you will see they will write capital A and small a. So this means it has an allele of A and A. This one is the dominant gene and this one is the recessive gene, right? So this means that there are homologous genes. Okay, so in order to match that homologous genes point by point, let's assume this chromosome is the gene for head structure. So another gene for head structure will come near it. Another gene for head structure will come near it. So this one is carrying the information for head structure. This one is also carrying information for head structure, but one is dominant, one is recessive. So now they will, since they are homologous, they are carrying the same thing, they will hug one another like this. From the front view, you will see it like this. They are hugging one another. And from the side view, you will see it like this. This one is from the side. This one is the front view. So this hugging one another, the process is called synapsin. And when they hug one another, it's like this marker. When you combine it with this marker, they become like this. So this structure that they form is what we call a tetrad. So at this process, then if you just look at your physical appearance, you will notice that you have inherited something from your mother and you've inherited something from your father. So how does that occur? It is at this process of this mitosis, of this meiosis. So after this tetrad is formed, now they will start exchanging information from one another, like these markers. So they will change their caps, like this. So So like they will change their caps like this. So this means when they get separated, this green marker will have blue cap and this blue marker will have red cap. So if it is this sperm that go and penetrate, definitely it will go with some parts, some information from the mother and some information from the father. If this is the sperm that got chance to penetrate, it will get some information from the father and some information from the mother. So this is how they do. So now if they hug one another, I told you they will exchange cap. So now it will become like this. From the side view and from the front view. From the side view it will become like this. So from the front view it will come like this. So do you get me up to this step? Alright, okay. So after this, now they will come and arrange themselves in the equatorial plane. So when they arrange themselves, they will now get, that is during the metaphase, they will arrange themselves. So after they arrange themselves, then this nuclear membrane will get ruptured and what do you call it? And the centrioles will come into play, creating microspindles. Assuming this is the inside the cell, the centrioles like this, they will create spindles since the chromosomes that are having different colors like this. So you have other centrioles from this side of the cell. Let me draw the complete cell. Assuming this is the complete cell. These markers are confusing me. Let me change their heads. Okay. So the microspindle will come out. And from here, cut this place. So now they have formed what we call the spindles. So after the spindles has been formed during the anaphase, now they will be separated from one another. So when they get separated, meaning this one will go this side and this one will go this side. So in the next cell, it will resemble like this. This is the centrioles. And then this is the chromatid. This is the chromatid. And from here also the same thing. So the same thing. So now they will be pulled apart. After the pulling apart at the at now the next step, which is the telophase, this cell will now they will now separate from one another, forming two daughter cells. One cell here, one cell here. It's having this type of chromosome. So this means since they were homologous pairs here, they arrange in one plane and they get separated from one another. So at this place we have a diploid cell. That is the mother cell is a diploid cell having 46 set of chromosomes. So because now they are separated, they are cut off into two, they will now have what? 23 pairs of chromosome, which is a haploid. So in mitosis, in what you call it, in meiosis 1, the key points are what? Formation of synapses, formation of tetrad, crossing over of when they hug they change caps, we call it crossing over, and then formation of haploid cells. So if you want to get, we'll continue meiosis 2 in our next lecture. So if you want to get access to some videos related to this embryology knowledge, you can go directly to our website www.drkamalmb.com. Thank you.